When we talk about research to the UG as well as PG student, we talk about central dogma of molecular biology. Under central dogma of molecular biology, we have steps like DNA is getting converted into the DNA, that is DNA replication. DNA is getting converted into the RNA, that is transcription. RNA can use the protein, that is a translation process. We try to explore this molecular biology technique in our biochemistry lab. So when DNA can be getting converted into the DNA, that is the replication process. We can show to the student the replication process with the help of polymerase chain reaction. So we have equipment required for this technique, that is replication. We have thermocycler and we have centrifuge machines. The related technique which are required for the showing the replication is present in our department. As well as when we talk about transcription, DNA getting converted into the RNA. So this transcription process we can do in our laboratory, that is gene expression techniques. For that we have real-time PCR machine. So with the help of reverse transcriptase enzyme, the consumable chemicals required, we can show the how gene expression can be done in the particular cells or the tissues. Then we have RNA to protein, that is protein expression. So protein expression we can do in our laboratory. So that is the flow of the equipments we have maintained. So basically we can do most of molecular biology lab work in our laboratory. We have UV transilluminator, then we have for detection Western blot equipment. So if you have some clinical sample and you want to detect for example any virus sample, rabies virus you want to detect from dog saliva, whether it is present or not. So we can use this PCR technique to identify whether that virus is present or not. Or otherwise, if you have particular antigen present in the infectious disease, you can detect this with the help of the antibody testing, that is Western blot. With the help of Western blot, we can detect that antigen. The clinical sample we can analyze with this one. So these are the different flow of equipment. So when we are going to see our laboratory, we have different equipment. We have ice flaking machine, which will be required for transformation or transfection to maintain the cool temperature of the bacterial cell or the animal cells, or any biochemical reaction which require the controlled temperature. That can be done with the help of this ice flaking machine. Then we have high precision electronic weighing balance, which helps to measure the precise chemical weight. Whenever we measure the chemical weights, so during that process, we have try to keep the related chemistry chart in our laboratory, like chemical structures or periodic table. So whenever UG student comes to me and he asks me how to calculate the molecular weight, I show him the molecular weight periodic table, and then from periodic table they get understanding how 1 molar NaCl can be calculated. So based on the molecular weight of sodium and chlorine, we can add it into up to the 1 liter and we can make 1 molar. So likewise, so basically one technique we are doing, the related charts we try to keep it. When we say water, or when we say alcohol or glucose, we try to demonstrate the molecular structure of that chemical entities. So for that purpose we have molecular kit with us. Means the molecular models we can generate, the carbon we can join, and we can make the glucose for them, or we can make the water molecule for them, or alcohol molecule for them. So this we can find in our laboratory how this can be done. When we say we have to incubate the bacteria or particular tissue sample, we have this water bath with us, or bacteriological incubator we have with us for handling the infectious agent, or to maintain the biosecurity protocols. We try to do everything under the laminar flow. So we have 2 laminar flow equipments in our laboratory. For culturing the bacteria, we have refrigerator shaking incubator. For separating the individual biochemical components, we have this centrifugation machine, cooling centrifuge machine. So if you want to do the bacterial streaking for showing its biochemical characteristic of the microorganism, we have the loop and the stirrer, this equipment, and whenever any infectious agents or any biological entities are handled, likewise serum or any bacterial sample, so in that case we have autoclave with us. So this with the help of that autoclave, we try to keep everything sterile. So before the experimentation and after the experimentation, all things should be kept in the sterile environment. So these things we try to maintain. That is the sterility of the process. So very important for the students as well as the instructor point of view, we try to maintain. When we see our biochemistry lab, we try to maintain the instructions for the students. So the visual instructions like how you should behave in the lab. So you should wear proper apron, we should wear the shoes, we should have the glasses on when you are handling the biochemistry equipments or the chemicals, because we are dealing with the dangerous chemicals sometimes, like concentrated H2SO4. So during this time, you should have the face shield, you should have proper gloves, and then you should handle this equipment. How you should handle these equipments. You should not keep the heating things on unattended. Any experiment, we should not eat anything in the laboratory. This instruction we try to convey to the students. So these are the important things in the biochemistry laboratory. And during the so this is about our PG laboratory. So basically under the PG laboratory, we try to provide the services to all college level students. So most of PG or PhD students comes to our laboratory. They comes from either the PCR or the serological analysis or any experimental techniques which involve the biochemistry parts. They comes to our laboratory. So we try to provide the services. In addition, so the next thing we have is in the TVCC, Teaching Veterinary Clinical Complex. In that one we have the clinical biochemistry laboratory. Under that clinical biochemistry laboratory, we have fully automated biochemistry analyzer. Next, the hematological analyzer, that is also fully automated. Next, blood gas analyzer, and then urine analyzer. So talking about the fully biochemistry analyzer, the purpose of the biochemistry analyzer is a serological testing. So we have total protein, albumin, globulins, cholesterol, AST, ALT. These estimations, for UG student, we do it by the handheld pipetting. But for maintaining the accuracy and the consistency in the result and to avoid the manual error, which can be done when the person handles does pipetting. So in that case, we have fully automated biochemistry analyzer. So around the 50 tests can be done in the 1 hour with that biochemistry analyzer. So that's this we have intentionally kept it at the clinical position, so that whenever clinical animals come in the clinic, the students, internship student or the postgraduate students of the clinical subjects which are available there, so they can do the analysis there with the help of the fully automated biochemistry analyzer. Next, we have hematological analyzer. So for hematological analyzer, the student can take the sample from the clinic and directly they can place the sample with this anticoagulant added to that machine, fully automated machine, and it can give the whole sort of parameter like RBC, WBC, the differential leukocyte count, so platelet count. Likewise, parameter can be given with that equipment's fully automated biochemistry analyzer. Next we have is a blood gas analyzer. So animals comes with a different complaint like acid-base imbalance. So whenever there is a respiratory acidosis, respiratory alkalosis, or the metabolic acidosis, metabolic alkalosis, acidosis or alkalosis, in that condition, it is very essential that you measure the amount of the which gases are in excess or the lower. That is whether there is a higher partial oxygen pressure or higher partial or CO2 pressure, or what is the pH, what is the electrolyte concentration in the blood. So instantly student can take the blood and directly they can feed the blood into the machine, blood biochemistry analyzer, blood gas analyzer, and they can analyze this with the blood gas analyzer, the amount of gases and the electrolyte and the pH there. Next, animals urine can be analyzed with the urine analyzer. So we have strip based urine analyzer present there. You put the strips in the urine and you feed that to equipment, and all your urine parameters can be detected with the urine analyzer. So basically the biochemistry, when we talk about our biochemistry component, we have one animal. The animal is eating something, some biochemical changes is happening in its serum, like protein level and other things, cholesterol. It can be detected in the UG laboratory, undergraduate or our clinical biochemistry laboratory. So the animals blood we test animal serum, we test animals blood gases, then we test urine. So most of things we try to cover under the biochemistry, being the basic department. So our biochemistry department comes under the basic sciences. So basic sciences, we try to maintain the basic flow. We try to maintain the flow from the genetic level, DNA level, to the to the molecular means microscopic level to the microscopic level, molecular to the macroscopic level. At the molecular level we study the DNA, at the macroscopic part we study the proteins which are present in the serum or the different metabolites. In addition, department is currently trying to have the HPLC equipment. This HPLC equipment, with the help of that equipment, the metabolic analysis can be done. So this metabolome analysis can be done with the HPLC equipment. And for UG students, some very basic things we try to maintain at our place. Like many students, they will be requiring the distilled water in the research. So how to get the distilled water? So getting the distilled water with the conventional technique is very tedious process. Means we have to heat this double distillation things. So for basic experimentation purpose, we try to maintain this molecular biology grade water for them. So each day they can take 15 or 20 liter water from our department for their experimental purposes. So we try, in that way the department tries to serve the UG student, PG student, PhD student, as well as the farmers which comes to our clinic to help them in the processes, means learning process, either the learning process or the animal care process. So in addition, these are the different. So basically we have maintained a lot of equipment repository with us, with the knowledge repository in addition to that. So when we maintain this repository, to our point of view, we try to display, we don't try to keep that equipments and knowledge with us. Equipment point of view, we try to give the equipment services to the students as well as the farmers. And knowledge point of view, we try to organize the trainings for undergraduate students. We have organized the DBT Star College schemes. So already, DBT Star College scheme. So under this one, we show the equipments, how equipment working can be done, how these things can be done, means experimental processes can be done, as well as we try to train the faculty, all India level faculty, with the help of the biochemistry training. Already we have conducted 2 10 days biochemistry training. Though basically for this training we try to get the faculty from the other places. So the most of all India level faculty, they were involved as a guest faculty in our training process, and they have very nicely delivered the lecture. So I am thankful to them also for their contribution to this training programs also. So in that way we try to do our part in the services of the students and the farmers. And
बोलो, काय प्रॉब्लेम?
पॉज घेऊ थोडासा.
हां, पॉज घ्या.